Welcome to our presentation on gene therapy and disease development. Lesson outline. In this presentation, we'll cover the following topics: definition of gene therapy, types of gene therapy, how gene therapy works, its importance in treating genetic and acquired diseases, mechanisms of gene therapy and its applications in disease treatment, discovery of DNA. The discovery of DNA by James Watson and Francis Crick in 1953 laid the foundation for modern biotechnology. Understanding DNA's structure and functions paved the way for advancements like genetic engineering, gene editing tools, and DNA sequencing technologies. Learning outcomes. By the end of this presentation, you will be able to identify the uses of modern biotechnology, including gene technology. Describe some of the practical and ethical considerations of modern biotechnology. Definition of gene therapy. Gene therapy is a medical treatment that involves altering or manipulating the genes inside a person's cells to treat or prevent disease. Its goal is to correct or replace faulty genes, introduce new or modified genes to combat diseases, or silence genes contributing to the disease process. Gene therapy applications. Gene therapy holds promise for treating various conditions, including genetic disorders like cystic fibrosis, muscular dystrophy, certain cancers, and viral infections. Techniques include inserting healthy gene copies, editing genes with tools like CRISPR, and using viruses to deliver genetic material into cells. How gene therapy works. Example, CRISPR. Let's take the example of CRISPR. The process begins with detecting and removing a faulty gene using a cutting enzyme. A synthetic defect-free version of the gene is created. The Cas9 enzyme cuts the faulty gene out, and the synthetic gene is then placed back into the DNA strand. Gene therapy involves modifying genes within a person's cells to treat or prevent disease. It has various applications and shows potential in treating numerous disorders. Primary uses of gene therapy. Gene therapy is applied to genetic disorders such as cystic fibrosis, muscular dystrophy, and sickle cell anemia. Cancer through techniques like CAR-T therapy and genetic modifications targeting cancer cells. Infectious diseases such as HIV and hepatitis B. Neurological disorders like Parkinson's and Alzheimer's. Autoimmune diseases like multiple sclerosis and type 1 diabetes. Blood disorders including thalassemia and Fanconi anemia. Regenerative medicine for tissue healing and bone repair. Developing DNA vaccines to prompt immune responses. Types of gene therapy. Somatic gene therapy. This type of therapy involves altering genes in non-reproductive cells, also known as somatic cells. The changes affect only the individual and are not passed on to future generations. It is commonly used for treating genetic disorders such as cystic fibrosis, cancer, or other inherited diseases. Types of gene therapy. Germline gene therapy. This therapy involves modifying genes in reproductive cells, such as sperm, eggs, or embryos. These changes can be inherited by future generations. However, it is not widely used due to ethical concerns and safety issues. Gene therapy has the potential to revolutionize medicine by targeting the root causes of genetic disorders, cancers, and other diseases. It offers hope for long-term personalized treatments. Thank you so much for taking the time to watch this presentation on gene therapy. I hope you found it informative and engaging.